Hello, students. Have you ever noticed how leaves look so fresh and lively in the morning sunlight, but by late afternoon, they sometimes appear a little droopy? That's because deep inside each leaf are tiny pores called stomata, and their behavior determines how efficiently the leaf exchanges gases and conserves water. Stomata are minute openings, mostly found on the lower surface of leaves. Their primary job is to let in carbon dioxide, which is essential for photosynthesis. But here's the catch. If stomata remained open all the time, plants would lose huge amounts of water through transpiration. So they act like natural walls, opening when carbon dioxide is needed and closing when it's not, striking a delicate balance between food production and water conservation. Isn't it fascinating that something so small regulates processes that keep plants alive, and us too? Now, have you ever wondered why leaves don't photosynthesize at night? That's because stomata have their own daily rhythm. In darkness, when photosynthesis cannot occur, stomata generally close. This minimizes water loss, which is critical for the plant's survival. But once the sun rises, stomata reopen. Carbon dioxide begins diffusing in, while oxygen and water vapor diffuse out. This coordinated exchange supports both photosynthesis and transpiration. So, every morning as you see plants turning toward the sun, their stomata are already busy, opening up to let in carbon dioxide and kickstart the process of food preparation. Let's zoom in a little closer. Each stoma is flanked by a pair of specialized cells called guard cells. These are no ordinary cells, they are uniquely designed with a thick inner wall facing the pore and a thin outer wall away from it. Guard cells are special because they contain chloroplasts, enabling them to photosynthesize. This allows them to control their own turgidity. Here's how it works. When guard cells become swollen or turgid with water, the outer walls stretch and the inner walls bend outward, opening the pore. When they lose water and become flaccid, the pore closes tightly. So the secret of stomatal regulation lies in these tiny but powerful guard cells, which literally act as gatekeepers of gas exchange. Now, let's look at the modern scientific explanation, the potassium ion concentration theory. During the daytime, the chloroplasts in guard cells carry out photosynthesis and produce ATP, adenosine triphosphate. This ATP is then used to actively pump potassium ions, or K ions, into the guard cells from neighboring cells. As potassium ions build up, the concentration inside the guard cells becomes higher than outside. This makes the guard cells hypertonic, so water rushes in by osmosis. The guard cells swell, become turgid, and bend outward, opening the stomatal pore. At night, the reverse happens. Potassium ions leak out, water flows out, the guard cells lose turgidity, and the pore closes. Before scientists discovered the potassium ion mechanism, there was an older explanation called the sugar concentration theory. According to this view, during photosynthesis, the guard cells produce glucose. This increase in sugar concentration raises their osmotic pressure, pulling in water from surrounding cells by osmosis. As the guard cells absorb water, they become turgid and bulge outward, opening the stomatal pore. Though this theory is not the most accepted today, it highlighted the importance of osmotic changes in stomatal regulation. In fact, it set the stage for the more accurate potassium ion theory we now rely on. But what about closing? Stomata close whenever water is scarce or transpiration is too high. When the water content of leaves drops, water flows out of the guard cells. They lose their turgidity and become flaccid. The thick inner walls then come together, sealing the pore tightly. This mechanism prevents further water loss, protecting the plant from wilting under stress. So, closing stomata is just as vital as opening them. It's nature's way of ensuring plants don't lose too much water while still carrying out photosynthesis when conditions allow. So let's bring all this together. Stomata may be microscopic, but their role is monumental. By regulating the entry of carbon dioxide and the release of oxygen, they make photosynthesis possible. Their opening and closing are controlled by guard cells through ion movements, osmotic pressure, and turgidity changes. The potassium ion concentration theory is the widely accepted explanation, but the older sugar theory also provides valuable insights. Most importantly, stomatal regulation maintains a balance between two critical processes, capturing carbon dioxide for photosynthesis and conserving water for survival. Isn't it incredible that such tiny pores, invisible to the naked eye, control the very breath of plants and, by extension, the breath of life on Earth? I hope you now appreciate just how beautifully coordinated these microscopic gateways are. In our next session, we'll see how stomatal regulation fits into the larger carbon cycle, connecting plant life with the entire biosphere. Until then, keep observing the green world around you. Every leaf is a laboratory of life in action.